Today on Futures in Biotech, Dr. Tommy Nilsson talks about the test run for the technology that will transition us into the next era of medicine. A special guest host, subject matter expert, Dr. Marc Gerstein.
Netcasts you love.
From people you trust.
This is Twit. Bandwidth for Futures in Biotech is provided by Cachefly at cachefly.com.
This is Futures in Biotech 79.
This episode of Futures in Biotech is brought to you by Netflix. Watch thousands of TV episodes and movies streamed to your PC, Mac, or TV instantly. Plus, get DVDs by mail in about one business day. For your free 30-day trial, go to netflix.com/twit.
I believe that biotech is the next frontier.
Probably the greatest intellectual revolution that's ever taken place in man's history. DNA is the code for life. We're actually beginning to understand how life works, which I think is something that's mind-blowing in and of itself.
There was going to be a genetic component to aging.
How long was the extension?
About 30-40% for humans. That would equate to something like 20 to 30 years.
How close are we to actually having a therapy of something?
Ballpark, 10 years.
It's potentially one of the things that will end up rocking the world the same way that people said, oh, the sun's the center of the universe, so this and that and everything. And now here's somebody who can come out and say, hey, look, here's how we compare it to our closest evolutionary relative.
Welcome to Futures in Biotech. I'm Marc Pelletier. Today, we are very fortunate to have two of the top guys in their field, and that is the field of proteomics, bioinformatics. Our guest is Dr. Tommy Nilsson. He's a professor in the Department of Medicine at McGill University and Canada Research Chair in Proteomics and Systems Medicine. He was recently on Futures in Biotech with John Bergeron. They did a show together, and they really set a case that it's time to do proteomics to revolutionize modern medicine. We're really at a transition between eras of medicine. One where we make guesses based on our parents' history, to one where we have a molecular anatomy down to the atom of who we are and our disease states. And to help out with the interview, I brought on a subject matter expert. Let me warn you that this show is going to be pretty hardcore biotech, and I don't think we should be scared away from it, but we should embrace it and try and get it even deeper. So to do so, we brought Marc Gerstein. He's the Albert L. Williams Professor of Biomedical Informatics. He's a professor of molecular biophysics and biochemistry and of computer science at Yale University. Welcome, guys. Welcome to the show.
Thank you.
Thank you.
So we've got a little bit of a lag, a little Skype lag, and feel free to interject anytime. We're in Canada, we're in the East Coast, and I'm in the Midwest, and we're piping through Petaluma, California. So while we're not trying to interrupt, it's definitely good to interject, and especially when the topic is going to be proteomics. And proteomics, let me just sort of put a little preamble here. If we can understand the human anatomy down to the atom, and you can compare a diseased kidney from a non-diseased kidney, a cancer kidney with a non-cancer kidney, and you can find those biomarkers very, very early. You basically have a tricorder. If you had a tricorder that could do that, you could do personalized medicine. Which drugs would work best for that exact disease state? And there was a paper that was written, and I'm going to put the paper up here, right here. Here's the title. It's a paper authored. First author was Alexander Bell, who he worked with them a little bit in John Bergeron's lab, and Tommy was an author on the paper with John Bergeron as well. And what they did is they sent out test samples of proteins to 27 labs, and I will stop there. It's the title of the paper is a HUPO test sample reveals common problems in mass spectrometry based proteomics. So I will ask you the first question. Let's just make it a little lighter. Tommy, how would you like to present this, and where does it stand today?
To go back to your introduction, which I think is both futuristic in a sense, but also I think depicts quite where we want to be in 10 years. You talk about the tricorder to understand the human anatomy down to the atom. Yeah, I mean it's for the purpose of curing disease, but I just want to make a distinction on, or give you an idea on how we approach this, because you up until recently have tried to find drugs that work that you can apply in disease context that work in a general population. But what you want to do, and using genomics, proteomics, and other omics techniques, is to use this to enhance the phenotyping of a patient. So imagine the doctor, the patient, and instead of just relying on blood sample, urine sample, imaging techniques, you could now bring the full force of the DNA sequencing, deep DNA sequencing, transcriptomics and proteomics and metabolomics to enhance the picture of that particular patient. And by doing that, the doctor is going to be much better positioned to make a informed decision about your treatment, and also the prognostics, the follow-up, etc. that comes afterward. So I think this is a very exciting time that we are in, where we can see the how these technologies can be applied. But I also then want to be cautious. And this is why I suggested this test sample paper, because that really sets a sort of benchmark in the field where we are today. Because one should temper enthusiasm with reality, and having the long-term objective clear. It's fantastic. It's exciting, but we have a lot of work in front of us to get there. So before I dwell into the aspects of the test sample study here, I return back to you.
Yeah, so I think one thing I know that it might be, we've done a couple of shows with you and John Bergeron, and then we did a show with Rudy Aebersold, three giants in the field, and but it'd be good right here to sort of revisit what exactly is mass spectrometry, so that people can see how this potentially could be used in diagnostics and visualize it. And maybe if you could give us a brief introduction to that, and then it'll set the stage.
Yeah. So the distinction, I think it's most people, they know it. It's easy to understand our how to say genetic makeup. We have chromosomes in every nucleus, in every cell in the body, and this encodes the proteins that are within the cell. So in genomics, you can sequence the DNA, and you can look for differences between different individuals, and there try to make links to disease. And this is exactly how the genomics field is advancing. Now, most drugs and treatments, they are not directed against genes, they're directed against the gene products, the proteins. And there is currently no real good correlation between the expression of the genes as you can measure on an mRNA level and the protein makeup of the cell. They have a vastly different turnover and so forth. So really what people are trying to get to is to be able to determine the protein profile of a cell or a subpart of a cell. And the way we do that technically is to have a protein sample. That can be a blood sample, serum sample, plasma sample. It can be a tissue extract. It can be part of a cell, like an organelle or enriched fraction of something that you have brought down with an antibody, etc. And now you want to determine the protein composition of that. So the first thing you do is to, and this is not exclusively, but the most common approach is to cut up the proteins into peptides using a proteinase, trypsin, and then you will have these peptides going into the mass spec, and the mass spec is a very sensitive instrument, can determine the mass with very, very high accuracy of these peptides that come in. And you can also collide these peptides and fragment them further down to smaller fragments, so that you can do what is literally boils down to the sequencing directly in the machine of these peptides. But the most common...
Let me interject just a quick sec. Can you move your camera down just a bit? Editor...
Sure.
Let's just mark this spot even a little bit more, so that the lower third is your chin is not touching the lower third. Pull your screen towards you. Keep going.
Yep. Okay.
Nice. That's better. Perfect.
Okay.
Yeah.
Good. So,
is that okay? Perfect.
Continue.
All right. I was just asking our engineer. All right. Cool. If that was good, if there was a good angle, so that's better. So you were saying that you have your peptides going to the mass spec after trypsinization, and then.
And then, so the mass spectrometer is an extremely accurate instrument, so it can determine the molecular weight of these peptides or any other compounds that you put in, for that matter. And by determining the molecular weight of the peptide, you can then go into a database and search where you for in silico derived peptides of the corresponding molecular weight. And in this way, you can then identify the protein from where these peptides have been derived.
How precise a molecular weight can you get down to an atom?
No, well, you could get down to 1,000 ppm, etc. And it's...
No, I meant not as an atom, but what's the smallest thing it can measure? Not in terms of quantity, but say you had a lot of hydrogen. Could you measure a lot of hydrogen, or what's the cutoff?
No,
electron?
So we go down to below femtomole in concentration of the peptides, and then you have, depending on the instrument, the mass accuracy will be, I think, around 1,000 ppm.
So this is Marc. I just wanted to chime in for a second. So I'm very interested in this process, that you use to identify things. I'm just curious, can you think about the process without a database search? I mean, can you just imagine using the mass spectrometer and just reading out the proteins without doing that database search? Is there any way to think about that?
Sure, but you could literally collide the peptides in such a way that you can get from that the peptide sequence. Before the modern approaches were used to do something called Edman degradation, and that was literally peptide sequencing. But every peptide sequence that you derive, you have to correlate to that to something. And that what you correlate to is a protein or predicted protein coding sequence. So unless you had that, you would basically have something that you would be completely in the dark with. So you have to, at some point or another, go back to a database of predicted sequences.
So if you chew up that protein and you take this one protein, it's hemoglobin. Let's use that as an example, because you're doing a red blood, a blood sample. You got hemoglobin. You can't just throw the hemoglobin into the mass spec. You digest it into little pieces, and those little pieces are, we know because of the human genome, certain sequences of amino acids. You got like a string of amino acids that connect to another string of amino acids that forms the full protein. You've digested into its subparts, And so you might have a 5 amino acids chain, a 10 amino acid chain. And based on the sequence that we have from the genome, you can make a prediction as to the exact molecular weight. Mm-hmm. And then the mass spec has a readout, and you try to correlate your predicted molecular weights of hemoglobin to the actual detected.
Yes.
That makes sense? Oh, okay. So I got it right.
So this is Marc. I think that I'm quite interested in that, but I'm just curious, what if there's something that, we haven't identified in the genome or that's producing a protein? Is it possible somehow to see that in the mass spec, or is that just sort of some sort of peak that you can't account for or something?
So in I guess in 99.999% of the cases, these peaks will not be looked at. But in the context of the proposal for the Human Proteome Project, and based on the outcome of the sequencing of the human genome, we now know that there are 20,300 predicted open reading frames. So with that, I think we are going to see less and less loss of peaks that we are not able to assign. But at the same time, you have to realize that we have a lot of post-translational modifications that we can't predict. And we also have variances between individuals, so-called SNPs. So these are single nucleotide polymorphisms, which then can translate into changes in the amino acid sequence. So sometimes you're going to be thrown off the mark by these type of variances. So it's not an absolute in any way method.
Oh, okay. No, I was just curious to ask those. I have some more on that, but I'll let you keep going.
Mm-hmm.
So go ahead. But I'm still amazed. I'm at here. I'm thinking just from the clinical side, you've got, you can do tests, you can push the liver, look for a reflex, look for a breathing response. You can listen to a heartbeat. You can take some tissue out and look under a microscope. Now medicine's going to the next level. I mean, you can sequence the DNA and make a prediction as to the likelihood of the onset of cancer. But what if you could actually see the atoms in the human body? And this is pretty much what it does. I mean, we're made up mostly of proteins, right? Proteins, lipid, sugars, all which can be, quantified or potentially quantified on mass spec. And so I really have to, take a step back from 37,000 feet here. This will change how we're diagnosed. And I've been going to the ER for gallbladder issues, and they're doing their blood tests, and I've got like 6 results, right? And I'll give you an idea of what's going on, but man, would it be neat. Fortunately, it's banal. But anyway, go ahead.
The technology is there. It's how we apply the technology. But you have to be also realistic in the sense that if you take a physician today working in a hospital and seeing a patient, he or she base their decisions on, I think now, today, it's 90% based on laboratory tests and imaging and so on. So that means that every physician will have to have some deep knowledge or insight into, or at least appreciate the technologies from where you derive these data. And if you now bring in genomics, it's going to increase that. It's going to make the life for the physician more difficult unless we can build up a very robust support structure that makes the interpretation of these data easy for the physician. So it's not just the technology challenge, it's also a translation challenge on how you can translate information to a physician in such a way that he or she can make the right decisions. So there's a huge challenge in the bioinformatics, also a huge challenge in how to integrate data from different levels, and then you come into the realms of systems approaches, etc. So proteomics as such, very powerful, but we need to combine this with other type of fields also, in particular with bioinformatics.
Would you say that the Human Genome Project, its real benefit of having done it, its real raison d'être, as you might say, is to enable proteomics, right? Genomics was good. You could watch genes be turned on, turned off with gene chips, but really, as laying the groundwork for proteomics, is it, or am I going too far to say that? And maybe Marc will get mad.
Well, actually, this is Marc. I wouldn't get mad there. But one thing that I think is interesting to think about, and this was why I was asking the questions earlier, is that I think people know that only a small fraction of the human genome, only about maybe 1% or 2%, is devoted to coding region, and there's a really, really large fraction of the genome that's not thought to code for proteins. And on the other hand, a lot of it's transcribed, and there's a very big debate about the degree to which that's creating RNAs, or the degree to which maybe there's some proteins or some translated products that we haven't found out there before. And so I was very curious about the ability of proteomics to try to find things that we hadn't already mapped in terms of the genome.
So I mean, to give you some sort of a sense on what can be done. I mean, the more we can map, when you do mass spectrometry, the usual approach is to do something called LC-MS-MS. So the peptides are then separated by a liquid chromatography. They enter the mass spectrometer, and you do what is called a parental scan. And then you get the peptides as they come in, and you get the flight of time, and you get the charge, and you can determine the mass of the peptide. Now, but the peptide from the same protein, the same peptide, is going to have the same behavior from time to time. So the next time you do it, the peptide is going to behave exactly the same. So you're going to be able to train your mass spectrometer, not the mass spectrometrist, but the mass spectrometer, to select those peptides that are of interest, and you select on the peptides for further fragmentation, and that further fragmentation then gives you the composition of that peptide in much more detail. So let's say now that we ignore everything that is there that we already know with high certainty the identity of the peptides. So now we can go in and look at these interesting ones that are not, coming out that easily, and then we can select those, and we can fragment them, and we can go deeper, and then we can determine the composition of those peptides and what protein these correspond to. So yes, it is possible to do that type of exploration with mass spectrometry.
One question I have also, and I hope this isn't too technical, but I think it's kind of an interesting question. When people look at the transcriptome, so the before you get to the proteome or looking at the proteins, people often look at the level that the genes are expressed, and as you pointed out, there's not that much correlation sometimes between the levels of gene expression and the amount of proteins, but people often look at the transcriptome. One of the things they do when they look at this is they sequence it, and often with this next generation sequencing, and they figure out when they sequence it, what fraction of it they can kind of map back onto the genome. And one thing people talk a lot about is the fraction that they can get mapped and the fraction that doesn't map. And then they spend a lot of time thinking about, well, the fraction that doesn't map, what do we make of it? Is it real? Is it an error, and so forth? And I'm wondering if is there some statistic they have in proteomics that gets a sense of, like, when you look at the entire protein repertoire in a cell, what fraction we can kind of account for, and what fraction is, we don't really exactly know what's going on?
So for that, I can give you, I can't give you an absolute number because it hasn't been determined, but I can give you a sense. So most people in the field of proteomics doing mass spectrometry, they will deposit their data into public databases. And there has been one study, it hasn't been published yet, but it's quite telling. So about 50% of all mass spectrometry data is against 86 proteins. And now you go up to about 90% of the spectra that are in the databases are about 2,000 proteins. And then about 95% of the spectra are against 6,000 proteins. And then it doesn't go higher than that. So you can then get a sense of this is the where we are in terms of the technology, etc. Because it's easy to see the abundant proteins, etc. Maybe we looked at easy tissues. We haven't looked everywhere in the body for these proteins. But I would say about more than half of all the proteins that you would predict has not been seen by mass spectrometry. And then if you look in the whole field, not just in proteomics, but taking everything else that has been done for the last 100 years, we're still going to be in the dark for, I think, at least 40% of all proteins have never been seen. Predicted proteins have never been seen. And there's only a scant knowledge about the majority of the proteins that have been seen. So we're really at the very beginning of understanding the biology behind the proteins, and we have to go in a systematic way to identify the proteins, to see them either by mass spectrometry or by antibody or whatever methodology you can apply.
No, that's very interesting. I mean, one thing that I think is particularly I'm particularly keen on is when they're annotating the genome. They certainly look at the transcripts and what they call it RNA-seq and so forth, or RNA sequencing. There's some people, and maybe you're one of them, who are then connecting these transcripts up with protein identifications and kind of feeding that back to the people who are creating gene structures on the genome. And I think that's I'm quite involved in this latter process, and so I'm very interested in how the proteomics kind of fits into this whole thing.
So with the paper that we have in front of us, the test sample study, the real wake-up call, the outcome of that study was that the databases that are...
Sorry. Let's do a cliffhanger. Sorry, maybe that's the worst way to present a cliffhanger. Sorry, Tommy. I gotta get... Leo's so good at this. Let's talk about the paper after we have to do a quick spot for Netflix, and then we'll come back and talk about the outcome of the paper, which is really, really cool because it sets the groundwork for transitioning out of the biotech labs into the hospitals, and it's really neat. By the way, here's a quick spectra. You get these sharp peaks. What we have, it's pretty hard to see. The graphic is pretty lousy, but we're looking at atomic masses. I mean, down to what's the final?
I can't see.
Oh yeah, it's kind of looking at a traditional. I mean, we're down to one proton in its ability to detect two different peptides by a weight difference of one proton. About anyhow. So let's get this spot done because this is supported by Netflix, and so I'd like to thank Netflix for sponsoring Futures in Biotech. They deliver movies directly to your home, and that saves you time, money, and hassle. You can instantly watch thousands of TV episodes and movies. They're streamed directly to your PC or Mac, or can be streamed to your TV via Netflix-ready devices, including the Xbox 360, PS3, Nintendo Wii. At our house, we use the Roku box in our downstairs TV. We use the Wii upstairs. We have an Apple TV, iPad, laptops, iPhones, iPods. Everybody has Netflix in our house set up on their device. So you can absolutely watch it any which way you want. We also get DVDs, and they send you DVDs by mail in about one business day. Watch as many movies as you want, anytime you want. There's never any late fees or due dates. My pick of the week, do you guys watch Netflix at all? Do you have time?
Yeah, no, I do. I have Netflix. This is Mark.
Yeah, it's really cool. So we were supposed to pick, do a Netflix pick, and I picked Battlestar Galactica. I'm a huge sci-fi fan, and this is created by Glenn A. Larson. It's a 2004 to 2009 series. They did 4 entire seasons. There's partial seasons as well that are added in, like I think there's season 3.5 and 4, 3.5 and 4. They did 75 episodes. So for 2 solid months, I watched nothing on TV but Battlestar Galactica. I missed out on all the main shows, excluded everything. I'm catching up on The Office now, but so they have it up on their instant view. So you can set up an instant queue and just whenever you want, on demand, go and watch Battlestar Galactica. So if you want to watch Battlestar Galactica or choose from any of the thousands of TV episodes or movies, you can when you register for a free trial membership. You go to netflix.com/twit. Do you have a favorite movie that's on Netflix, Mark?
Oh, me, a favorite movie. Well, I saw this one called Babies recently. It was it was all about, I think it's called Babies. It was all about babies in different countries. Very good movie.
Cool. Has Netflix come to Canada yet, Tommy?
Sorry? Yeah, of course. Yeah.
It has.
I don't know if this movie is on Netflix. It's the progenitor of most sci-fi movies. This is called Forbidden Planet. And that's the one I have as a favorite because it really predates every sci-fi movie, and it's with a Canadian as a lead character, Leslie Nilsson, who is
he's great. He's great.
Yeah. So but he's not funny in that movie. He's very serious.
Let me search it. What's the name of the movie?
Forbidden Planet.
Yeah, it's there.
Okay.
Let me see. Is it on Instant? I'm not sure it's on Instant. Oh, no, but you can get the DVD, Forbidden Planet. It's a pulp fiction sci-fi classic, Forbidden Planet. Stars Leslie Nielsen as a heroic starship captain. All right, well, I'm going to put it on my queue. So they're going to send it to my house. That's really cool. Hey, I'm glad it's up in Canada because it'll. I travel to Canada when I go to my in-laws, and it's often, fun to, when you're kind of on pseudo vacation, to really get into it. So I'm done with the Battlestar Galactica series, so I'm moving on.
Canada is not exactly as it is depicted in South Park.
Right. Or yeah.
This is Mark. One thing that is worthwhile to mention about Netflix is I'm not so much a watcher of it, but Netflix has made a tremendous impact on the machine learning community. And we talk about it all the time because they have this thing called the Netflix Challenge, which we really think is quite amazing. I don't know if you've heard about this, but it's a challenge that Netflix put out to all the people who do computer prediction algorithms to try to essentially predict who would watch what movie. And they put $1 million prize out for it. And it's been quite, I think it's fair to say, quite inspirational for a lot we
Wow. Did you have a submission? Did you?
I think one person in my lab did submit. We certainly didn't win, but we certainly watched the competition. There's websites where you can watch the different teams involved, and it made a very serious impact on the I would say the machine learning community.
Let me just say, Mark, there's another way to do it, and we are in the United States. You give me a call. We'll form a corporation. We'll patent the algorithm, and we'll approach Netflix for a licensing deal. Okay, and you guys get the entire ownership of the company. I'll help you out that way, and you can make more than $1 million if you have a great algorithm to help predict what people are watching. So give me a shout. We'll get it financed. You're in New Haven. There's a lot of great VCs that would love that opportunity to have some of your insights into algorithms as well. So we'll talk after the show. Be sure to sign up for your free trial at netflix.com/twit. We thank Netflix for the support of Twit and Futures in Biotech. I hope I didn't offend you though, because I can understand how there's being a pioneer and then there's doing a company, and you've had a great contribution as a pioneer in the field of bioinformatics.
Oh no, I think it was a great idea. Actually, a lot of people, a lot of people thought about how they could get that $1 million or something. So I think that's a creative way to do it.
Yeah, but you can, oh man, if you can predict how people are going to behave, I mean, they've got a great suggestion, suggestions for you, movies to suggest, which are great. But if you can do an even better job, it's worth more than $1 million to Netflix. I think they're 20% of primetime TV watching now.
Wow.
Well, but I digress. Let's get back to, I guess changing modern medicine and transitioning from microscopy, from blood tests that are based on antibody recognition, to getting down to the atomic level. Tommy, you were talking about the HUPO test sample study and its outcomes. So this is a 2009 paper where they sent out test samples to see how effective a mass spec would be and to debunk the sort of the way.
So to give you a little bit of a backdrop, the field of mass spectrometry based proteomics has had its sort of moments where you have had a lot of claim to fame, and that then has fallen flat on its face. So there has been then a lot of discussions, a lot of there have been a lot of promises and very little delivered at the end of the day in terms of biomarkers and value for the community. And the European Commission in 2006, I think it was, really asked HUPO and the field of proteomics to come up with ways to move forward, to standardize the field, to set basically standards, and to prove that the technology and the field could deliver. And so the one part of that process has been then to conduct different type of test sample studies, and this has been done. This one was done through HUPO. There has been other test sample studies done by the ABRF and also some other private or individual initiatives that have been carried out. But this
and
one,
HUPO is the Human Proteome Organization?
Yes. So that's the equivalent of the HUGO, which is behind the human genome. So this was then a request. And we then went forward to think, okay, how can we make a test sample in such a way that it is pure proteins in there? Because one of the objections in the past, and one of the problems in the past, has been, yes, there's been test samples sent around, but then people find all sorts of things in there, and so then they come back and say, well, this is not really fair. You're asking me to identify proteins, but you're sending me a lot of rubbish. So a humongous effort went in to actually ensure that the proteins that were contained within this test sample were what they was supposed to be, so they were purified extensively. And if you happen to be able to go into Nature Methods supplementary material, I think there are about more than 50 pages documenting the purification and characterization of each one of these 20 proteins that went in, and how they were selected, and so on.
So you guys were really setting up the framework, not for looking for a needle in a haystack, but here's the haystack, guys. Can you see it? Right?
Yes. This is a famous, I would say, analogy is that there is an elephant in the room and no one is seeing the elephant, right? So it's like that. So, but we wanted to make sure that no one was going to come back and say this sample is full of rubbish. So these 20 proteins were then painstakingly purified and put in equimolar amounts. These are all human proteins, equimolar amounts, into one test sample. And so the first task was then to identify these 20 proteins. And then the proteins had been chosen in such a way that each protein had a peptide which had a molecular weight of 1250 Dalton. So the task number 2 was to identify that peptide and show that you could select that peptide and identify it in each one of these 20 proteins. Now, these test samples were then sent out to 27 participants, mostly academic labs, and also a couple of mass spectrometry vendors that wanted to participate. And the result was awful. 7 labs managed to identify the 20 peptides, the 20 proteins. And the rest, they failed in some way or another. And only one lab out of the 27 could complete task number 2 to identify the 1250 peptide. So,
that's not encouraging.
This was a catastrophe. So of course, when you do something like this and you have the result, which is then, I would say, at the very least, a very surprising result, then you have to ask the question why this happened.
Let me just ask you a question here really quickly, because you said 1250 Dalton peptide. So there's this one peptide that you throw in there to make sure that it's like a nice control.
It's part of the protein.
Was it part of all 20 proteins, or is it 1250?
Yeah. So when you digest with trypsin, you are going to generate a 1250 peptide. It's not the same peptide, but it's going to have the same molecular weight.
Okay, and so it's sort of gives. Go ahead, Marc.
This is Marc. When you say it's a 1250 peptide, it doesn't. You have 20 peptides that are approximately the same weight, right? You can actually separate them, right?
Yes.
Yeah.
So
let me ask one last question. Sorry about this. We got this lag, and I talked about it a little bit before. But so 1250 Daltons. What is a Dalton? It's 10 to the minus 27 kilos. It's the mass equal to 1 proton or a neutron, right? So what an incredible instrument if it can detect, these slight differences of you've got a sample, a peptide sample. That peptide weighs 1250 Daltons, and you're able to decipher between slight changes within a few Daltons. That's just outstanding. 10 to the minus 27 kilos.
But that's what the instruments do, and that's
sure. They just do it.
But also you could just take the open reading frame from a database and ask, give me the 1250 peptide, and that's what we did. To select the proteins, so you can predict this also. So that's not it's not in any way close to tour de force to do this. This is what you would expect to be able to do on a routine basis.
Sure, it's like saying I'm going to travel at light speed. It's not hard. You guys are.
This is Marc. I'm just curious. So I don't know that much actually about exactly how this type of thing would be set up. But is it fair to say that you have these 20 proteins, they fracture into lots of peptides, and you wanted to make sure that at least one peptide from all the proteins was in kind of the same area, so that you could kind of tune the instrument to the same rough 1250 sort of region and see it. Is that what's going on?
It's not exactly. So we wanted to try to simulate a more complex situation. Maybe if you compare the first task to a walk in the park, the second one would be to sit down on a bench and open your bag with a sandwich and eat it, and that's really the level. So you wouldn't necessarily need to run the mass spec to predict the 1250. So if you identify the protein, then you should be able to look at the reading frame and say, okay, that's the 1250 peptide coming out from there. So it's not a humongous challenge to do that. But the abysmal performance is then very, how do you say, telling in that only one lab managed to report back on these 20 peptides. And you should remember that if you look on the paper, these are labs that have had agreed before to be co-authors on the publication. So you have to then assume that you take this seriously because you are conducting a study. It's going to be published here in Nature Methods, which is a kind of it's a high-profile journal, and therefore you're not going to just throw this and not bother about this. You have to assume that people made an effort and try to complete this task.
So this is Marc. So if that's the case, why did it come out with such discrepancies?
So this is now the interesting aspect. So what you call, but before I get to that, I also want to say then that during the test sample study, of course, the first author of the paper, Alex Bell, who was conducting this study, and also the only person who knew which lab was reporting in data, etc. He was the only person that actually could link the data to the particular participants. He then at various points reported on the status of the study, and invariably the criticism was then that the test sample had to be contaminated and rubbish, right? So there was a lot of talk, rumors, discussions, etc., trying to explain away the dismal outcome of this study. So what we then decided to do was not just to rely on the reporting of each one of the participants, but we also requested that each participant would submit their raw data, the mass spectrometry data that had not been analyzed, the raw data, to submit that. And then we could pool all the raw data. We could do something which is called a centralized analysis, and then see what did the mass spectrometer really show for each one of these participants. And in the majority of the cases, they had fantastic data. It was absolutely no problem whatsoever. So they failed miserably in connecting the dots between having the raw data from the mass spectrometer to connecting that to the database, and from there to the identification of the protein. And that's where the majority of everyone failed. So
this is Marc. I'm just curious, when you're saying that the data was very good, the actual spectra, like they ran the machines, and the actual spectra from all the machines, they looked at it, and the spectra were similar. It was just the interpretation of the spectra that was different?
Yes, the matching of the data to database, that's where it failed. So it really showed that the mass spec technology as such is really, really good. I mean, there was one or two participants that had some problems with the sample, but overwhelmingly, it was really pristine data that came out of the mass spectrometers. And it didn't really matter so much. There has been discussions about having standard operating procedures, etc., to standardize, etc., but people were not adhering to that. They were just doing it the way they thought would be the best way to do it, and they generated fantastic mass spec data. It was then using the search engine and the database, and that's where everything crashed. So that was an immensely valuable lesson.
That was a very intuitive Here's a Spectra, by the way. So that was very, very intuitive to say, you know what? I bet mass spec is ready. I think it's just, I mean, this is not a hypothesis driven experiment. This is, I mean, that's a huge finding, but how did you predict that? I mean,
we didn't predict. This was born out from the criticism which happened at conferences, that we must have sent out really rubbish samples. We must have sent out samples that were contaminated with keratin and God knows everything, right? And this is why people had a problem then to analyze them. But by bringing in the raw data from each participant, we could actually see ourselves what had the participant observed with their mass spec by analyzing their raw data. And we could prove that some labs introduced themselves keratin, etc., which is a very common contamination in protein samples, and so forth. We could also really accurately determine the amount of bacterial proteins which were there. We capped that at 6% because each protein had been produced in bacteria. So they were 96% pure, each one of the proteins. But we had no contamination from any other source in this test sample. But the revelation that came, the answer to why they had failed, that was really brought out from necessity to take the raw data and analyze it, because people were trying to explain away the result of this test sample by claiming that the test sample as such was rubbish. So call it serendipity or just, but we didn't predict this, but it was very satisfying because it really for us showed, and also for the community, showed that mass spectrometry is a very powerful technology. You just have to be very careful in how you apply it and how you then try to match the spectra to the database.
But I'm curious about these database discrepancies. I mean, can you give a sense, like an intuitive sense of how people can differ in their database lookups, or why this would happen? I mean,
So in this case, in this test sample study, we provided a frozen database so everyone was using the same database. They were free to use the particular choice of search engine and so on. But the database was the same. Of course, in real life, you have a couple of different databases you can use for your annotation, but in this case, everyone used the same. But it really highlighted a big problem because, and I can just give you one example. I mean, right now in most databases, although there is an enormous effort, and I recognize that, to curate the databases, there are still multiple entries for one particular protein, for instance. I mean, just take any protein like transferrin receptor. You will have transferrin receptor, and you will have transferrin receptor like protein, and so on, so on, so on. And you will have maybe 10, 20, 50, 100 entries for a particular protein, and they may differ one base or more. But this doesn't help when you're doing mass spectrometry, because you really need to be able to identify the protein, meaning it should be transferrin receptor at the first level, and then once you have made that identification, you then can go down to the detail on what isoform it is, what SNP it is, etc., etc. But right now, that's not feasible. And therefore, people have constructed software that collapses these long, multiple entries into one. But that's software, and that's not fail-safe. What happens if you really want to do it carefully is that you have to do manual curation of your data, and that is a very, very tedious process and can take months to do.
So this is Mark. So you're saying that, like, on a first level, the discrepancies were there was 10 of these, say, transferrin receptor type entries in the database, and one person would report the first entry, another person would report the third entry, and so forth. That type of thing. But that's the type of thing where you kind of could, after the fact, see what happened and kind of put them into register. Is that fair?
Yeah, I think there wasn't that. How should I phrase it? It wasn't major tweaks that needed to be done to get this into a shape that is how do you say, something that we can live with. But it was a necessary process, because we could really pinpoint what the real bottlenecks are in the annotation. And as a consequence, maybe not completely as a consequence of this, but certainly I think it contributed a lot. Now there are protein-centric databases being constructed, like the knowledge base of Amos Bayrock, for example, the Swiss prot derivative, which is he calls the NextProt. So there you really go in and you have one protein. And of course, there are many different forms of that protein, but it's one protein. And therefore, I think with that type of structure, your ability to hit the protein from your mass spec data is going to be improved.
Okay. Well, this is Mark. I mean, it just sounds, I mean, from what you're saying, it sounds like it doesn't, even though it's upsetting in the sense that the labs didn't agree that well, it does sound, this sounds like a very fixable type of thing.
Absolutely. But it does, it highlighted something which, and I think, I think a lot of people in the proteomics could draw a sigh of relief, because we didn't know this. We didn't know this before. We thought either people are completely useless or they are making up data or whatever, because it was totally all over the place. But now we could pinpoint it to something which I think in retrospect seems trivial. But it isn't. It isn't trivial, because we need to fix this. And I think it's easy to fix.
Yeah, well, one thing I'll just say, I mean, just in defense of the databases, and obviously this is something I know a bit about, I do think it's very non-trivial to do that. And actually, I think just because I suspect people would want to get a sense of this problem in a more simple way, I'll just say that the issue is that, you have this concept of a protein, but as you were saying, it has many different potential alternative splices. It has many SNPs in many different individuals, and maybe people have studied many various sequence variants of it, or where it's truncated and whatnot, and kind of collapsing all of these together. It's actually a rather tricky thing. Picking out the thing that you will then call the canonical version of the protein. It's not a trivial thing.
I fully agree. But my training before I went into biology is from engineering, and I look at this from an engineering perspective. And if you have 20,300 open reading frames that are predicted to encode proteins, you can predict the proteins from that. And you can then also go in and identify the peptides that are what we call proteotypic. So this is a huge effort from Rudy Aebersold and also other people in Seattle, like Eric Dorch, etc. They have now big databases on peptides that are prototypic. That means that a peptide, if a protein is there, it should generate this peptide. And you then empirically also see whether or not this peptide is able to ionize and fly in the mass spectrometer, so that you can correlate that. So that database is there and is growing, and we can then with higher accuracy really say that, okay, with this peptide, that means we see this protein. So of course then you can go one step below that and say, okay, now I'm not just interested in identifying the protein, but I also want to see what particular isoform of the protein. And then you can go in and look for isoform-specific peptides within that context. But it's all built around a gene-centric or protein-centric approach, where there's one protein having a lot of different variants, but it's one protein, and it's called whatever. If it's transferrin receptor, it's called transferrin receptor, and it's not called Bob or Bill. It's called transferrin receptor, and just that simple fix will make life so much easier for anyone who's trying to annotate their mass spec data.
It seems like it would be a very good tool for discovery as well. If you could go with, you've got your known knowns, then your known unknowns, things that you predict are there based on the genome, and combinations of all the post-translational modifications, either a glycosylation, sugar added, or a lipid added, phosphorylation, which is a phosphate group gets out of the protein. So that those could be predicted to happen. So you got your known unknowns. But I guess at the question in the end is the unknown unknowns, the unknown knowns, the unknowns unknowns. Sorry. So you got your known knowns, your known unknowns, and your unknown unknowns. Right, to quote a famous CEO.
But this is it's not going to be like we're doing a mass spectrometry analysis and then that's it. This is like any other discovery, biology, etc. Is that we are going to discover more. We're going to discover more isoforms and so forth, and learn more and more. Sorry,
Mark.
I'm here.
Okay, there's some sound coming in the stream.
Okay, so we are gonna improve our knowledge, and by improving our knowledge, we also improve our ability to predict which peptide should be seen for a particular protein or isoform, and in that way, you can then go in, and that opens up another avenue, which is then to be able to do absolute quantitation by using reference peptides.
Has those 27 labs that were analyzing these test samples, have they come since 2009 now to unify their workflows such that they can start working on that unification of methodologies such that could potentially be translated to clinic? Or is this just way too early?
This is. I hope everyone has drawn from the outcome of this, but it's not just the labs per se, but the whole community, and also the community of bioinformatics and databases that have seen that this is something that we really need to move towards, to enable mass spectrometry-based proteomics to be more robust. And that's a prerequisite for even thinking about applying this in the clinic. But there are serious efforts on their way to do this. I have no doubt that in a couple of years we're going to be there. Because the fix is not that enormous.
This is Mark. I think this is an interesting thing to point out, in the sense that, I think one question one can ask is, if it's not that hard to do this, why hasn't it been done? And I do think it points to an interesting situation where I think it's easy to describe, in a sense, what to do. This is this sort of simple idea of, you have 10 versions of a part, you want to find a canonical part name for it. But to do it right on a very large scale for many, many things is actually a very difficult undertaking. And I think one reason that probably it hasn't been done properly is people haven't appreciated how difficult it is, and probably haven't afforded the appropriate credit to the people trying to do it. You know what I mean? So that there's an appropriate incentive for people to tackle it. I suspect it's one of these things where no one has really put effort into it, because it didn't seem like it was sufficiently rewarding.
Let me add to that, Mark. I think we're witnessing it. And this is the whole point of Futures in Biotech, is to be at that frontier at least as witnesses to some of the great transitions of science forward and how it affects technology, right? So we're witnessing it now. It is happening. These mass specs are out there. People are trying. They're coordinating, and this was a beautiful example of a coordinated effort to bring that technology to fruition. And so, sometimes we are at that frontier, and you got to recognize, holy moly, we are at the frontier. How come nobody has done it? They're doing it, and we can see it happen. And that I feel really fortunate to be able to discuss it with you guys.
So there is a little snippet that I can maybe add to all this. So there has been commentaries written about this test sample, and in some cases the conclusions have been made that it's only highly qualified labs that are able to do good mass spectrometry-based proteomics. I just want to say that is we're not allowed to disclose the data linking them to the participants, but I can say as much that there is no correlation between whether or not you're a well-established lab or not in how the results came out. So that's a misconception that is actually. Sorry.
Can I ask if Paul Predke's group got it right? Did Predke?
I'm not going to reveal any names, not whatsoever. All I can say is that the lab that got the more difficult task done was not a very known lab. So my point is that there was no real correlation between being highly established or not, whether or not you perform well or not. And I think this is again points back to the robustness of the mass spectrometry technique. But at that point in time, the weakness of the linking the mass spectrometry to the protein identification, and it's still a lingering problem, but we are dealing with it.
There's a question in our chat room. Titus said, "Isn't fast protein liquid chromatography cheaper to separate peptides rather than mass spectroscopy or mass spectroscope?" It's part of it, right? Or yes. You use MS instead of, sorry, not MS. You use liquid chromatography to separate the peptides before it enters. That separates the FLC, separates the peptides into their constituents that you throw into the mass spec.
Yes. And different peptides, they have different binding capacities to different matrices, and so they will enter the mass spec depending on your liquid chromatography setup, then at different times. And if you use the same column and the same settings, etc., you can then do this in a very reproducible manner. And the technology now is switching to using high pressure liquid chromatography machines, and you can then get a much better, you can use much longer columns, like 1.5 meters, and then get very, very good separations between the peptides, and then you enhance the ability to detect and identify the peptides. So these are called UPLCs.
Well, this has been a really amazing opportunity to have this discussion at this stage, right? And what I'm trying to do here, we're approaching our 5 years of Futures in Biotech now. And if you go back and look at the list of shows we've done several series of shows in various fields. And we've just taken on proteomics this year. But this is a beginning. And we're going to follow it over the next, hopefully, next 5 more years, and maybe be able to witness this paradigm-shifting technology and how it affects not only medicine but the human condition, because we're taking our ability to see the human anatomy to the next level, which is just so cool. I think it's really neat.
It is cool, but at the same time, inject a bit of modesty. You're gonna have this together with other technologies. It in no way replaces. It's not a standalone technology. You have enormous power in imaging techniques. You have enormous power in genomics, in transcriptomics, in metabolomics, etc. And this will be one facet of being able to understand what's going on in the cell, in the tissue, in the organ, in the human body.
Oh yeah, it's not going to replace. Definitely not going to replace. I mean, MRI is just absolutely fantastic, and that's organismal level, but and maybe some tissue level, but and microscopy is getting better and better with laser scanning microscopy, or even some forms of tomography used in an electron microscope, transmission electron microscope. You can take a protein sample and tilt it back and forth and actually solve the structure of the protein, sometimes with a single protein.
But what we all need, and I guess I'm there directing this to Marc, is that we all need the very needed support from the bioinformatics community, from the systems approaches that are being developed in systems biology and systems medicine, etc. And for all the people and all the hard work going on in trying to curate these databases.
So we should we should cover this again. We should do it perhaps when would be a good time? Probably in about 6 months, and see if how much closer we got to the clinic. It'd be a fantastic opportunity. So I'd like to thank our guests today. Were there any final thoughts? I think we've got it. I mean, there's a lot to digest here, and I'm going to listen back again. Marc, do you have any last questions?
No, I had a great show.
All right, cool. So thank you to our guest, Dr. Tommy Nilsson. He's a professor in the Department of Medicine at McGill University and Canada Research Chair in Proteomics and Systems Medicine. I'm really glad that you're moving this forward. You're going to certainly help us in our old age when we get diagnosed, and quality of life is improved.
I gonna grow old before that, perhaps, but we'll see. We'll keep optimistic.
Well, thank you very much for coming on. And
thank you for having me. A pleasure.
The state of the science. And I'd like to thank you also, send a thank you to our special guest host, Marc Gerstein. He's the Albert L. Williams Professor of Bioinformatics, Biomedical Informatics, Molecular Biophysics and Biochemistry, and Computer Science at Yale University. Thank you very much, Marc, for coming on. Your enthusiasm for science is just unbounded, and it's amazing to participate in discussion with you. Thanks for helping out here.
Thanks for inviting me.
We're hopefully going to have you back on, and we're going to talk about your work on possibly June 24th. We'll awesome. We'll coordinate. Yeah, you've got a lot of really neat stuff since your last episode. And I mean, Marc does some great stuff. He even goes into the genome and mines for our history and our genetic history. So he does some pretty incredible stuff. And a lot of the technologies that in the area of bioinformatics have come from Marc's work. And so we'll talk about it in June. I'd also like to thank Burke McQuinn for handling the audio and video boards and the recordings today. Thank you, Burke. I'd also like to thank the team that make this possible at the Cottage: Leo Laporte, Lisa Kensal, Frederic Louis, Eileen Rivera, Tony Wang, Mike Taylor, John Salina, Jeff Stewart, Jason Howell, and Dick DiBartolo, and the rest of the team in Petaluma, California. Lastly, I'd like to thank Phil Pelletier and Will Hall for the opening and closing themes. I'd also like to send out 2 shout-outs. Julia Wynn, she's a Harvard student. I'm pretty sure she's still a Harvard student. She's in her final year. We'll learn more. She teaches a summer class at MIT that was inspired by Futures in Biotech. So she even calls the lecture series Futures in Biotech. So I'm hoping to have her on as a guest host to host an episode on synthetic biology, one of her main interests. So Julia, send me an email so we can touch base, and we'll to let me know if you finished your classes, and we can get started on producing that show. Also, shout out to Sharon Doring. She's a novelist, and she's working on a biotech thriller. I don't think it's quite out yet, and I just want to let you know that I haven't forgotten. I'm just a really slow reader. So far, so good, though. So I'll send you my comments very, very soon. Any comments or suggestions, you can reach me at Marc, M-A-R-C, at twit.tv or on Twitter at Marc Pelletier. Futures in Biotech. I'm Marc Pelletier.